Hey everybody, so today we're going to go into something new called enzymes. So before I go into explaining what enzymes are, just one term I want you to be familiar with. It's called a catalyst. So what's a catalyst? So basically it's an agent that provokes or speeds significant change or action. So a person that quickly causes change or action. So that's catalyst can be anything, anything in life. So a catalyst can be a reason for a decision or choice you make. Right. So same with enzymes. Okay. So enzymes are proteins that function as catalysts. All right. So this means they help to speed up chemical reactions. So in our body, there's all kinds of chemical reactions taking place, and one of the most important ones is digestion. Okay, we'll talk about it in a little bit. So this means that they help speed up chemical reactions. How? So they lower the activation energy of reaction, so it takes less energy for the reaction to take place. Enzymes are very specific and will only catalyze with one specific chemical reaction. Okay, so this means there are tons of enzymes at work at all times. Okay, so the enzyme won't change after the reaction takes place. All right, it's recycled, so we never lose them. So enzymes and chemical reactions. So we're going to go a little bit into what exactly happens, how they react in our body. So they happen in all living things continuously, and that's so important. Why? For stuff like breaking down food, and for plants when they do photosynthesis, right? Which is a means by which they make their own food. All right. So basically, this all happens, right, with the help of macromolecules, and they're called enzymes. Yes, they are macromolecules because they're mostly made of proteins. And as we already know, a macromolecule, right, is a protein. Okay, so up on top we have the reactants. So these are two chemical compounds that are forming, right, to make sucrose. Now, we want to break that down. Okay, so we add sucrase in this case and water. Sucrase is the enzyme, and enzymes always end in an ASE. Okay, so this one is sucrase. And so sucrase and water, they react, and they help to form glucose and fructose, which we know are what? Sugars, right? That's the product. Okay, so we have the reactants, and then we have the products. And enzymes help to make those come about. So enzymes, like I said before, they're so important in digestion. So they help break down large molecules of food, right, biomolecules, and make them into soluble ones, which can be absorbed in our small intestine, right, so that they can be absorbed into our body through the bloodstream. And different parts of the digestive tract have different pH conditions. Don't worry, I'll talk about pH a little later. And those help to enable different enzymes to work. So there are enzymes in our mouth, and then of course in our liver. Okay, there's a substance called bile. I'll talk about that later. But we have them in our stomach. Okay, and then the large and the small intestine. Okay, so we need those to break down food that we take in to help provide energy, right? Because we need good energy to function. So, like I said, the mouth, okay, it all starts there. And then down the esophagus to the stomach, all right? Once it's in the stomach, that's where there's a lot of acid, highly concentrated acids. And so some of you may have had, unfortunately, where you've thrown up. Has it ever felt like your throat just burned so bad because all that stomach acid? Okay, and that's all the enzymes breaking down all those proteins in your stomach. Okay, so you have a lot of acids in your stomach, and then of course we have the liver. The liver makes a green liquid called bile. All right, that's stored in the liver, and it's secreted to help break down fats. And then in the small intestine, okay, that one right there, okay, absorbs food. Okay, so that one breaks down food and it absorbs all those nutrients. But then in the large intestine, right, that's where water is being absorbed by the body. And of course, number 8, out the body, right? There's enzymes there too. So these are just some of the names of particular enzymes. So the first one is amylase. That's in the mouth, right? The salivary glands. Then we have protease. That's in the stomach. All right. Then we have the liver, the bile salts, the pancreas, amylase, protease, and lipase. And then the small intestine, maltase and protease. So remember, enzymes always end with an ASE name. All right. Remember that. So now we're going to go to the lock and key model, which is the shape of an enzyme, which allows it to do a specific job, kind of like a lock and a key. So there's a specific enzyme that joins to a specific substrate, which you're going to see in the next slide. Okay, so here we have a perfect example. So we have the key going into the lock. So we have the substrate and the enzyme, so they fit perfectly. All right, and then in part B, right there, boom, they join together like pieces of a puzzle. Don't worry, this is gonna make sense in a little bit. So the structure of the enzyme, okay, we have the enzyme and the substrate, and they both fit together so that they match like a puzzle, and that way it's activated. So why do we need these things activated? Okay, that's the next question. All right, so we talked about the lock and key. So the molecules that bind to the enzyme are called substrates. All right, so we have the enzyme and the substrate. The location where they bind together, that's the active site. After the enzyme and the substrate bind together, that's called an enzyme substrate complex. So these substrates can break apart. Okay, breaking apart, that's called denature. So they can denature or bond together to form a product. Okay, an example is disaccharide turns into monosaccharides. So remember, this is where we're going back to talking about carbohydrates. Okay, so they join right there, and then they can break apart, right, or denature. And here's a little funny, right? So enzymes and substrates, it's like they have a little relationship going on. And again, you know, some enzymes and substrates fight over where they're gonna bind, right? So what is the importance of enzymes? Why do we need them? They're involved in almost all biochemical reactions in our bodies, and they allow reactions to occur at the rate necessary for life. In animals, an important function of enzymes is to help digest food, right? That's the most important one, because we need to break down that food in order to get energy, right? So digestive enzymes speed up reactions that break down large molecules of carbohydrates, proteins, and fats into monomers, right? Smaller molecules that the body can then use. So without digestive enzymes, animals or humans would not be able to break down food molecules, right? The biomolecules, to be able to provide energy that we need in order to survive. So the body needs sustenance, right? So what are some factors that affect the actions of enzymes? One is temperature. So the rate of an enzyme increases with temperature. If the temperature is too high, it can denature, right? So I talked about that earlier. Basically what they split apart, okay? And destroy the shape of the enzyme so it no longer fits in the substrate. Okay, so we're gonna talk about what is pH. So the pH scale ranges from 0 all the way to 14, and it measures the hydrogen ion concentration. What is that? That's acidity. Okay, it expresses the acidity or basicity of a solution, right? So we got acids and bases. So acidic, right? Think of an orange, pineapple. So all those things, as far as the acidic go, we need those in our stomach to help us break down all those macromolecules we take in. Okay, so what are some of the factors that affect enzyme action? Some more, okay? So remember, we talked about pH, all right? They work efficiently at specific pHs, all right? So enzymes in blood, we have them in blood, they work best at a pH of 7, which is kind of neutral, right? So there's not really, it's not too acidic. There's not much breaking down going on. But in the stomach, all right, we have a pH at 2. So that's way down close to the 0 on the acid scale. So that's very acidic, and we need that in order for us to break down food. And then enzymes in the intestine work best at a pH of 8. All right, so that's a little more of a base. All right, so we're not really acidic, not too much breaking down going on. Okay, so some more factors that affect enzyme action: chemical messengers, right? Hormones. They can signal a cell to start or stop an enzyme from working. So hormones have a big role also in what takes place in our body. An inhibitor may prevent or slow the enzyme right by blocking the active site where the substrate sits down. So an inhibitor can sneak in there and kind of shut things down, so there won't be any breaking down going on. So just for you to remember, all right, the things that affect the enzyme action: temperature, pH, hormones, and inhibitors. And then these are just some examples of biological enzymes that we have that we take in every day. Avocado, I love avocado. So lipase breaks down fats or lipids, remember those? And then protease breaks down proteins. Cellulase breaks down fiber, okay, which we find in a lot of fruit. And then amylase breaks down starch, right? We find that in carbohydrates. Lactase, oh my gosh, dairy products. Is anyone here lactose intolerant? I am. I drink almond milk because of that. So we don't have lactase that naturally occurs in our bodies in order to break down the lactose. And that's why when you drink milk and you get a real bad, like a stomach ache, or you feel sick or nauseated, just because of that, you're lactose intolerant. So if you are, I recommend you switch to almond milk, which I drink every day. And then we got sucrase, sugar, right? Break down sugars. And then maltase breaks down grains. So that's pretty much it. Next up, I'll explain what we're going to do next. Okay, good job, everybody.